Welcome back to this next video in which we are studying headaches. Okay, so we're currently in the process of studying the story of the pathogenesis of migraine headaches. And let me begin this video by summarizing where we have got up to in this story so far. So we started the story with an individual who suffers from migraine, and that means that this individual, for genetic reasons, their brain is susceptible to the phenomenon of cortical spreading depression. So, when we take this individual who suffers from migraine and expose them to the triggers for migraine headache, what's going to happen is that cortical spreading depression is actually going to occur on some portion of either their cerebral cortex or their cerebellar cortex. Usually the cerebral cortex, but the cerebellar cortex may be affected in some individuals. So the triggers will actually trigger the cortical spreading depression to occur, and it's the cortical spreading depression that is fundamentally the brain phenomenon that gives rise to the migraine headache. Now, different individuals with migraine are susceptible to having cortical spreading depression occurring in different portions of cortex. So the different individuals with migraine, they will be susceptible to having this cortical spreading depression in different regions of cerebral cortex. Now, this is the difference between migraine without aura and migraine with aura. In individuals with migraine with aura, which remember is a third of individuals with migraine, the portions of cerebral cortex that are affected, that are susceptible to being affected by cortical spreading depression, include the primary visual cortex or the primary somatosensory cortex or even the primary motor cortex, very, very rarely. And when those areas of cerebral cortex are affected by the phenomenon of cortical spreading depression, we have discussed how that will give rise to visual, sensory, or even motor auras. The individuals with migraine without aura, they are not susceptible to cortical spreading depression in those regions of cerebral cortex. The regions of cerebral cortex or cerebellar cortex that they are susceptible to cortical spreading depression occurring on are separate, and when the cortical spreading depression occurs there, it's not actually going to give rise to the same sort of neurological symptoms, so they don't get an aura. Okay, so cortical spreading depression, it's the fundamental phenomenon that underlies migraine, we believe, in both migraine with aura and migraine without aura. The difference is where it's actually occurring. If it occurs in certain regions, it can give rise to an aura, and therefore you suffer from a migraine headache with a preceding aura. If it occurs in other regions of the cerebral cortex, then it won't give rise to an aura, and therefore you have a migraine headache without the preceding aura. Now, how then does the cortical spreading depression phenomenon actually give rise to the headache? Well, it's going to lead to the activation of the overlying meningeal nociceptors. So, overlying the portions of cortex where the cortical spreading depression is going to occur will be meninges, and these will be innervated by meningeal nociceptors. And the cortical spreading depression phenomenon is going to lead to the activation of those meningeal nociceptors, and it's that part that I now want to discuss in more detail. I want to discuss the actual headache of migraine now in detail, the pathogenesis of that, and answer some of the crucial questions that we still have concerning that. We've dealt with the aura, we're now going on to the migraine headache. Right, so the first thing to explain again, and I have already explained this, but I'm going to explain this again just to make sure it's absolutely crystal clear, is that there is a delay between the cortical spreading depression and the actual activation of the meningeal nociceptors. And the reason we know that is because in people who suffer from migraine with aura, of course, the aura that precedes the migraine headache by up to potentially an hour, that actually corresponds to the cortical spreading depression occurring on the cerebral cortex at that very moment. However, the headache doesn't occur for some time afterwards. Indeed, the headache doesn't usually occur until the cortical spreading depression is just about ending, basically, and the neurological symptoms of the aura are going away. Therefore, we need to justify why is that delay? Why do the meningeal nociceptors not get activated immediately by the cortical spreading depression? Why does it take a lot of time, up to an hour, before the cortical spreading depression actually succeeds in activating the meningeal nociceptors? And I've given you our explanation for that earlier on in a previous video, but I'll say it again. we think that the way that the cortical spreading depression actually activates the meningeal nociceptors is by some chemical diffusing from the area of cortex undergoing the cortical spreading depression to the meningeal nociceptors, rather than it being the case that the actual activation of the cerebral cortex is what activates the meningeal nociceptors. So indeed, you might think, as I've said previously, that the way the cortical spreading depression occurring on a portion of cerebral cortex would activate the overlying meningeal nociceptors is by the overactivity. So remember, cortical spreading depression, you'll have a rim of overactivity that's spreading outwards. You might think it's that bit where it's overactive that will activate the overlying meningeal nociceptors, but that can't be the case, otherwise you would get the headache and the aura occurring at the exact same time. The instant the cortical spreading depression starts, you would start getting the headache and the aura at the same time if you were going to get an aura, and that's not the way migraine with aura works, so that can't be the way that the meningeal nociceptors are activated. Instead, we think it is the case that something diffuses from the patch of cerebral cortex where the cortical spreading depression is occurring to the overlying meninges, and that chemical, whatever it is, will activate the meningeal nociceptors. Now, that chemical does not necessarily need to be the same chemical that was actually causing the cortical spreading depression phenomenon by diffusing out over that portion of cerebral cortex. Indeed, when the cortical spreading depression occurs, loads of strange molecules go up in the extracellular fluid that should not go up in the extracellular fluid normally. And this is why it's so difficult to actually decide which one is the one that's causing cortical spreading depression and which ones are a result of cortical spreading depression. So it could be some of these other mediators that go up as a result of the cortical spreading depression that then diffuse into the overlying meninges and result in the activation of the meningeal nociceptors, rather than it being the actual chemical mediator that causes the cortical spreading depression that we believe is there. Okay, right, so that explains the delay, that we think something has to diffuse to the overlying meninges and that takes time, and that explains why there is a delay between cortical spreading depression, which corresponds to the neurological aura in migraine with aura, and the actual following migraine headache. Right, there is another question that we need to answer about the migraine headache though, which is, why does it last so long? The cortical spreading depression, this is going to lead to the activation of the overlying meningeal nociceptors, but I've told you that this thing will be resolved pretty much in an hour, just as the migraine aura is usually resolved within an hour. Remember, the aura corresponds to the cortical spreading depression. When the aura is over, that's the cortical spreading depression over. However, the headache that follows the aura can go on for up to 3 days or even beyond that. So why do the meningeal nociceptors remain activated for so long after the cortical spreading depression has ended? What keeps them activated? And I'm afraid the answer is that we believe there is a vicious spiral. I won't say a vicious cycle. A vicious spiral is the better way of describing it, because it gradually gets smaller and smaller and smaller until it eventually goes. But there is a vicious spiral that maintains the activation of the meningeal nociceptors for a much longer time, and that's what I now want to study with you. So, I've erased some of our previous drawings so that we've got some nice space here, and let's now talk about this vicious spiral. So this vicious spiral is by the name of neurogenic, and this seems like a very thick pen, but it's my normal thickness. This vicious spiral is by the name of neurogenic inflammation. This is what maintains the activation of the meningeal nociceptors for so long after the cortical spreading depression has ended. So let me explain what neurogenic inflammation is. Neurogenic inflammation is inflammation caused by the activation of nociceptors in a tissue. And the meningeal nociceptors, the nociceptors that innervate the meninges, they are capable of actually causing neurogenic inflammation. We don't think that all nociceptors in all tissues are actually capable of causing neurogenic inflammation, but those in internal structures, such as the meninges, they usually are capable of causing neurogenic inflammation. And let me motivate this. So, if the meningeal nociceptors become activated, what should that actually mean? That should mean that there is a big problem. The meningeal nociceptors should not become activated. You know, this is not a normal pain coming from the meningeal nociceptors. This is something very bad, most likely, because the meninges are not a peripheral tissue. Therefore, they're not exposed to the normal sort of stimuli that you might be exposed to, which will cause pain. When by normal stimuli, I mean, you know, if you if you prick your thumb on a spike, on a little pin or something, that's a sort of normal pain because that's damage to the peripheral superficial tissues. That's the sort of pain that we expect, and we expect nociceptors in the peripheral tissues to occasionally be being activated because of exposure to painful stimuli that are in the environment. But we do not expect the nociceptors in visceral tissues, deep tissues, such as the meninges, to be activated. If they have been activated, then there is something wrong in these deep tissues, and that is unlikely to be some superficial noxious stimulus that has caused that. That means most likely that there's something very wrong in the meninges, and therefore we need to activate inflammation. We need to bring in troops from the bloodstream to make sure that there is not some sort of infectious pathogen present within the meninges that has led to the activation of the meningeal nociceptors. So, this is the phenomenon of neurogenic inflammation, when the activation of nociceptors in a tissue can actually activate inflammation in that tissue. And I hope I've kind of justified why you might want to activate inflammation in a tissue. If the tissue has its nociceptors activated, then something is very wrong in that tissue, and when it's a deep tissue like the meninges, that won't just be that there's been some superficial damage. It will most likely be that there is something majorly wrong in there, such as a pathogen being present in there, and therefore we need to begin the inflammatory response. So that is why the meningeal nociceptors are capable of activating neurogenic inflammation. So now what I want to discuss is the mechanism underlying this. So for this, let me draw one of the nociceptors of the primary nociceptive afferent innervating the meninges. So here is the primary nociceptive afferent, here in yellow. That's representing its axon, its peripheral process. And here in red now, this is actually representing the nociceptor, the structure on the end of the primary nociceptive afferent, this tree-like structure, which is actually detecting noxious stimuli. And this will be present somewhere in the meninges. So, cortical spreading depression through some mediator coming from the cortex to the overlying meninges has somehow led to the activation of this. So we don't know what that mediator is. I'm afraid I can't tell you the answer to that, but we think it's probably some chemical mediator diffusing across to explain the delay. So cortical spreading depression somehow leads to the activation of this nociceptor. This is very worrying because this is not a peripheral nociceptor. This is not going to be activated because you've touched something that's too hot, because this isn't in a peripheral tissue. So when this has been activated, it suggests that there is something very worrying occurring. Most likely a pathogen potentially is present in the meninges and is leading to cell damage that's then leading to molecules being produced that are activating the nociceptor here. So what's now going to happen is this nociceptor is going to release molecules that can cause inflammation, and this form of inflammation is called neurogenic inflammation. So let's draw on these molecules. So the cortical spreading depression is going to lead to two things that this nociceptor is going to do. Firstly, this nociceptor is going to activate its primary nociceptive afferent here, and therefore the primary nociceptive afferent will fire action potentials up that will actually lead to the headache, but also it's going to be releasing pro-inflammatory molecules. Now, you might be expecting me to at this point say it's going to release interleukin-1 and tumor necrosis factor alpha, the classical pro-inflammatory mediators. However, no. Nociceptors, when they're participating in neurogenic inflammation, they release different mediators to the classical ones. So here are some new famous molecules. So calcitonin gene-related peptide, CGRP, is an example of a mediator of neurogenic inflammation. So let me write this thing's name out in full. So the C is for calcitonin, and then the GR is for gene-related, and then the P is for peptide. So this is calcitonin gene-related peptide, CGRP, one of the mediators of neurogenic inflammation, and it's a protein, as indeed this name suggests. It's a peptide molecule, just like interleukin-1 and tumor necrosis factor alpha are indeed peptides. The other mediator, big mediator of neurogenic inflammation, is SP, which stands for substance P. Again, this is a protein mediator. So, when the Meningeal nociceptors are activated, not only do they send action potentials up their primary nociceptive afferents, which is what will give rise to the initial headache, they're also going to release calcitonin gene-related peptide and substance P, and these are the reasons that the headache is going to go on for so much longer, because if they didn't release this and you were just getting the cortical spreading depression activating action potentials, then the moment the cortical spreading depression stops, or at least a while after the cortical spreading depression stops and the stuff that is diffusing through and activating the nociceptors has stopped coming, then the primary nociceptive afferents would stop firing action potentials, and therefore the headache would end. So that doesn't explain how this can go on for up to 3 days. So indeed, it's the substance P and the calcitonin gene-related peptide that are being released here that are going to lead to the prolonging of the headache here. So we release these two molecules, and these two molecules are going to cause inflammation in the meninges. So how are they going to do this? Well, they're actually going to act directly on the microvasculature itself. But before I discuss that, let me talk about the effect that it's going to have, these mediators are going to have on sentinel cells in the meninges. So let me put this over here. So, substance P and calcitonin gene-related peptide, they're both going to be present now within the meningeal layers, and they're going to activate the sentinel cells of the meningeal layers. Now, let me just talk a little bit about this, because you might not be used to the idea that the meninges actually have sentinel cells present within them, but they do. So all tissues pretty much in the body have sentinel cells present within them. And sentinel cells, remember, these are like guard cells. They're cells that are present amongst the normal cells of that tissue, and they're constantly on the lookout for signs of a pathogen, or signs that there is something majorly wrong in the tissue. And if they find those signs that there is something majorly wrong in the tissue, signs of a pathogen, then they set off the inflammatory response. They release the classical mediators of inflammation, like interleukin-1, tumor necrosis factor alpha, a special type of sentinel cell called a mast cell releases histamine, and other mediators like interleukin-6, prostaglandin E2, these major mediators of inflammation. So, calcitonin gene-related peptides and substance P, they are actually going to activate the sentinel cells. They are a sign to the sentinel cells that something is majorly wrong. And the meninges do have sentinel cells present within them. Now let me just tell you about the different types of sentinel cells. Now, there are 3 major different types of sentinel cells, which are dendritic cells, resident macrophages, and mast cells. And these are present in loads of different tissues around the body, including the meninges. So the meningeal layers, they do indeed have dendritic cells present within them, resident macrophages present within them, and even mast cells present within them. So, let me just draw a few little pictures here, of the different sentinel cells. So, we'll start off with the dendritic cells, which are a major example of sentinel cells, so dendritic cells. Dendritic cells for short are often abbreviated down to DCs, and the reason they're called dendritic cells is because they have a star shape, they have dendrites coming off them. So let me just draw a picture of a dendritic cell, and I'll change colour here. So they look like this. These are the dendrites. Now, they are absolutely nothing to do with neurons. These dendrites are just processes coming off the major cell body, and that's all the dendrite means. It does not mean anything necessarily to do with neurons. Of course, multipolar neurons do have dendrites coming off them, But these cells are not capable of doing what neurons can do. Even though they've got dendrites, they are not capable of conducting action potentials or anything like that. They quite simply do not have the necessary voltage-gated ion channels to be able to conduct an action potential that neurons do have. So they're not an excitable cell, so do not get confused by the fact that they have dendrites and that they're called dendritic cells. So here's a picture of a dendritic cell. Now the reason it has these dendrites is so that it can stretch far, far away from its cell body, and it can be stretching into loads of different parts of the surrounding tissue, and therefore it can be looking for signs of pathogens in a large area, or rather large volume, of tissue. So the dendrites, they just extend its jurisdiction effectively. They extend the domain of tissue that it can actually be standing guard over. So there's a picture of a dendritic cell. Then let's do the resident macrophages. So macrophages are a major cell of the inflammatory response. When inflammation occurs, they're a cell that we bring in a huge amount to the inflamed tissue, and they come from monocytes that are in the bloodstream. However, all the tissues around the body, they do have resident macrophages that are already present there, dotted around the tissue, and these are performing a sentinel role. So remember, macrophages have both a role as a sentinel cell and then a role as an actual cell of the inflammatory response. So the numbers go up hugely in the tissue following the inflammatory response, but they are present beforehand as well. So resident macrophages, and for short, macrophages are often abbreviated down to M5s, like so. Okay, so let me draw a picture of a resident macrophage. So they're great big blobs of cells. They're nowhere near as interesting to draw as a dendritic cell. They're just a great big blob like so. And again, they're performing the same role. They are standing guard, looking for signs that there is a pathogen. And of course, by signs and by looking, what I really mean is that they have loads of receptors on their surface for molecules that no human cell would ever in its right mind produce, that would be giveaway signs that there is some non-human cell present within here. For instance, loads of bacterial cells, they produce molecules that no human cell would ever produce. And we can have receptors for those molecules on the surface of our sentinel cells, and indeed we do have, those receptors, when they bind their ligand, will activate the sentinel cell to say, there is a pathogen present here, you need to initiate the inflammatory response, you need to release the mediators and tell the microvasculature we need help, and the microvasculature can bring in the troops. Okay, and the final major type of sentinel cell is the mast cell. And these ones are special because these have their own very fancy inflammatory mediator, a very famous inflammatory mediator. In fact, this is probably the most famous inflammatory mediator, which is histamine, made obviously incredibly famous by its role in hay fever and allergic reactions and indeed asthma. Okay, so let me put the mast cell in here, but also important in inflammatory responses all over the body, not just in the respiratory tract. So here's our mast cell. It's again quite a boring cell. It's a nice circle shape. It's not as big and blobby as a macrophage. It's a nice circle shaped cell. And of course, its cytoplasm is full of lots of little granules. So these are little vesicles called granules that will contain the histamine that it can release. And that's its special inflammatory mediator. The other two types of sentinel cells, macrophages and dendritic cells, they are not allowed to release histamine. They do not have that privilege. Mast cells are the ones that specially can release histamine. Okay, so we've now drawn pictures of the three types of sentinel cells. So all over the body, you will have these cells dotted around amongst the other cells, the normal cells of each of the tissues. And as I say, their role is to stand guard, and if they see signs of a pathogen, they activate and they release their pro-inflammatory mediators, which are the alarm signals, the alarm bells, to the microvasculature to tell them to initiate the inflammatory response. So now the way that neurogenic inflammation is working here, look, it's very simple. The nociceptor here has decided there is something majorly wrong in the meninges because indeed they've been activated to fire action potentials. They've then release these mediators, substance P and calcitonin gene-related peptide. These will go to the sentinel cells and bind to receptors on the surface of the sentinel cells and activate those sentinel cells. And they are a sign then that there is something wrong in this tissue, in the meninges, and now these sentinel cells are going to release their pro-inflammatory mediators just as they would if they'd actually found signs of a pathogen directly. Okay, so again, let's just recite out what the major inflammatory mediators are. So, the mast cells, firstly, they have a special one that no one else is allowed to release, and this is, of course, the famous one, histamine. So they're going to release histamine, but they're also going to release all of the classical ones as well, along with the macrophages and the dendritic cells. So all three of these sentinel cells now, they're all going to release the classical ones. And as I said earlier, the two classical ones that you must know the names of are interleukin-1 and tumor necrosis factor alpha. Other ones that we mentioned earlier were interleukin-6. I'm not going to mention that one now in the pathogenesis of neurogenic inflammation. It was very important back when we were discussing tension-type headaches. However, interleukin-6 isn't really that important now. Interleukin-6 is one that is very important in causing systemic symptoms of inflammation, very important in going into the blood and causing fever, very important in going into the blood and causing tension-type headaches. But it's not particularly important in actually initiating the inflammatory response there and then. Nowhere near as important as interleukin-1 and TNF-alpha. These are the ones that majorly act on the microvasculature and cause the inflammatory response. Interleukin-6 is important in going into the bloodstream, going far away and causing at far away tissues systemic symptoms of inflammation, as indeed interleukin-1 and TNF-alpha were also important in doing. But I'm not going to mention interleukin-6 here. So interleukin-1, TNF-alpha, another important one for us to know about is prostaglandin E2 as well, that they will release prostaglandin E2, and remember that is the cause of inflammatory pain. That is capable of, oh, and I've managed to put a horrible dot there, so do excuse me whilst I waste some time getting rid of that. So there it goes, off it goes, good riddance. Right, so prostaglandin E2 is the major mediator of inflammatory pain. It can activate nociceptors and cause pain, cause their activation, cause them to fire action potentials down the primary nociceptive afferent. So you might see how there's going to be a vicious cycle here, which is actually going to be a vicious spiral, because the nociceptors release these, they've activated sentinel cells to now release prostaglandin E2. Prostaglandin E2 can now come back here and activate the nociceptor again. When the nociceptor is activated, it will release more of these, and it goes round and round and round. And indeed, this is how the headache is maintained for so long by this vicious cycle that neurogenic inflammation is causing. However, we'll get there later on. Now, interleukin-1 and TNF-alpha, and indeed histamine, they are major pro-inflammatory mediators, all three of them. They act on microvasculature blood vessels and cause the changes in those microvasculature blood vessels that cause inflammation. Now, it's not just those three, however. Substance P and calcitonin gene-related peptide, they are also capable, as well as activating the sentinel cells, they are also capable of directly activating the microvasculature themselves. So this is why I'm now connecting them all up here for this arrow here. So all five of them, calcitonin gene-related peptides, substance P, histamine, interleukin-1 and TNF-alpha, they are now going to work on the microvasculature blood vessels. So by that I mean the terminal arterioles, the capillaries, the post-capillary venules, the tiny little blood vessels. They're going to trigger changes in the behavior of the endothelial cells of those blood vessels, and the changes that will overall lead to inflammation occurring. A bit of inflammation is going to occur in the meninges now. So you will get an inflammatory exudate produced, which will contain proteins that are coming in, remember, to attack pathogens, even though they're not existing in this case. And you'll also bring in more white blood cells. And those white blood cells that you bring in will include macrophages, but they'll also majorly include neutrophils. So you'll bring in those of neutrophils and those of macrophages, macrophages, both phagocytes that are coming in to phagocytose the pathogen even though it's not there. Okay, so all of those are going to work on the microvasculature to cause inflammation. So let me just finish my little flow diagram off here to say acting on the microvasculature is then going to lead to inflammation occurring in the meninges. So a little bit of inflammation is going to occur in the meninges overlying the area of cortex that has undergone cortical spreading depression. Okay, right, but the important thing for us is how this forms a vicious cycle by some of the mediators of inflammation, some of the molecules produced by the neurogenic inflammation, activating the nociceptors again. Now, I've just mentioned that prostaglandin E2 does this, but it's not just prostaglandin E2. Actually, histamine is also a major activator of nociceptors. So both of those two, histamine, and indeed I'll try and connect up prostaglandin E2 with my green line over here. So prostaglandin E2, both of these are now going to activate, and I'll go back over here, they're both going to activate the nociceptor, the meningeal nociceptor up here. And therefore, this is how we set up a vicious cycle. However, I would prefer you, rather than calling this a vicious cycle, call this a vicious spiral, because it is going to end. If it was a vicious cycle, it would just go on and on forever. Calling it a vicious spiral is better, because that is why it actually ends. It gradually gets smaller and smaller and smaller and smaller, the activation of the nociceptors. Okay, so let me summarize what we have learnt here, and then we'll see the big picture of how this unilateral headache, the unilateral migraine headache, is going to last for so much longer than the cortical spreading depression. So the cortical spreading depression initially happens, potentially causing an aura, as we've discussed. A little while after that, minutes to up to an hour, what the cortical spreading depression will actually result in is the activation of the meningeal nociceptors in the meninges overlying that portion of cortex. And the way we believe that occurs is by some molecule that is released into the cerebral cortex when it's undergoing the cortical spreading depression, diffuses out to the meninges, and this is going to activate those meningeal nociceptors, and they'll fire action potentials, and there will be the onset of the headache now. And it will be unilateral, because usually the cortical spreading depression is only unilateral. So normally the headache is therefore unilateral. And remember, if you do have an aura, usually that unilateral headache will be on the contralateral side to where the aura symptoms are, either in the visual field or the sensory field or the motor field. So, even once the cortical spreading depression is over then, why does the headache often continue on for hours and hours and hours? The reason is that when meningeal nociceptors are activated to fire action potentials, they also release these molecules, substance P and calcitonin gene-related peptides, which are the mediators of neurogenic inflammation. And neurogenic, by the way, means that it is created by neurons. It's inflammation that is caused by neurons, hence neurogenic inflammation. And indeed, it is being caused by the neurons here. And many primary nociceptive afferents, not just in the meninges, but in other visceral tissues, where if they're activated, it clearly shows that there's something very, very wrong there, because it's not likely that they've been activated just because of peripheral external noxious stimuli, like you've touched something too hot. It's not like that. Because these are deep tissues that are being innervated by these nociceptors, if they've been activated, the body interprets that to mean that there is something very, very wrong there. So neurogenic inflammation is not just a feature of meningeal nociceptors, it's a feature of nociceptor activation elsewhere in the body as well. So, the nociceptors will release substance P and calcitonin gene-related peptide. These are the two major mediators of neurogenic inflammation. There are others, but these are the two that, if you're going to learn any, these are the ones to learn. These are going to cause neurogenic inflammation in two ways. They do actually act on the microvasculature blood vessels directly to cause the inflammation, but very important for us to know, they're also going to actually act on the sentinel cells. Why is it so important for us to acknowledge this? Because the sentinel cells are then going to produce the histamine and the prostaglandin E2 that are important in the vicious cycle component. So, One of the major ways that they actually cause the inflammation is by activating the sentinel cells in the meninges, and we have all three types of sentinel cells in the meninges: dendritic cells, resident macrophages, and mast cells. They're all present in the meningeal tissues, dotted around the place. They're all going to be activated by the substance P and the calcitonin gene-related peptide, and they will release inflammatory mediators, and the important ones that I have named here, there are loads of inflammatory mediators. I could have given you a list that will go on and on and on for pages, but I've just given you the most important ones to know about. Mast cells, they have an exclusive one that they are allowed to release, which is histamine, and all three of the sentinel cells, they can release these three here: interleukin-1, TNF-alpha, and prostaglandin E2. Interleukin-1 and TNF-alpha, forever shall their names be revered, are the major pro-inflammatory mediators. They are very, very strong. If you put them into an area of tissue, it causes inflammation in that tissue. They act on the microvasculature very powerfully and cause the inflammatory response. So interleukin-1, TNF-alpha, histamine, substance P, and calcitonin gene-related peptides, all of those, not just interleukin-1 and TNF-alpha, they're all capable of doing this, activating the microvasculature and causing the inflammatory response. It's just interleukin-1 and TNF-alpha are the most powerful at doing that. They're very, very important that you know their names. Okay, right. The component of the, how does this form a vicious spiral? It's histamine and prostaglandin E2 being produced in the meninges, or being released in the meninges. These both can activate receptors on the surface of nociceptors. We know this about prostaglandin E2, I've just told you it's about histamine as well, and therefore they can activate the meningeal nociceptors again, and therefore it becomes a vicious cycle. This will cause pain now. This will replace the cortical spreading depression. Once this has ended, it's the histamine and the prostaglandin E2 that are going to be activating the meningeal nociceptors in that area and causing the primary nociceptive afferents to fire action potentials that will maintain the headache, therefore. And of course this is forming a vicious cycle because the meningeal nociceptors will now release more substance P and calcitonin gene-related peptides, and it will go round and round and round. However, it's a vicious spiral because it'll get gradually smaller as it continues going round. So initially, the cortical spreading depression will lead to massive activation of the meningeal nociceptors. They will then lead to substance P and calcitonin gene-related peptide being released, and then those will lead to histamine and prostaglandin E2 being released, and then now let's suppose the cortical spreading depression is gone, the histamine and the prostaglandin E2 will be responsible for activating the meningeal nociceptors, but they're not going to activate the meningeal nociceptors as strongly as the initial cortical spreading depression did, so the meningeal nociceptor activation will go down, and now the release of substance P and calcitonin gene-related peptide will go down, but it will still be there, so you'll still get more prostaglandin E2 and histamine being produced, but gradually everything's going to be spiraling down, so that the activation of the meningeal nociceptors goes down, the release of the neurogenic inflammatory mediators goes down, histamine and prostaglandin E2 go down, so gradually it spirals down, and that's the reason that this eventually does end. Okay, so that then is why the unilateral headache of a migraine headache can indeed last for much longer than the cortical spreading depression because of this vicious spiral process caused by the neurogenic inflammation. Right, so I have a few more things that I want to say about the pathophysiology of migraine headache. Firstly, I just want to remind you of where the headache pain usually is present. Just remind you about where it's usually referred to. Then I want to talk about why is it pulsating, and hopefully you should have a good idea of why it might be pulsating. Why did we discuss the arteries in so much detail? Then I want to discuss the accompanying symptoms, the nausea and the vomiting, the meningeal symptoms, and then we'll end by discussing cutaneous allodynia, and then we will have completed our discussion of the pathophysiology of migraine headaches. So remember where the pain is going to refer to, and for this let's just go back to our picture of the brain here. So, remember, the meninges covering the front portion of the brain, and in fact all of this portion that I'm now highlighting in the laser pointer here, all the way to the back, what's that innervated by? Branches from the ophthalmic division of the trigeminal nerve. Remember, there's the recurrent meningeal branch of the ophthalmic nerve, and then there's the anterior branches from the anterior ethmoidal nerves that innervate the front. So the trigeminal nerve ophthalmic division covers a huge amount of meningeal nociceptors, all of this sort of top and front and back of the brain, and indeed quite a lot of the bottom of the brain that faces down to the tentorium cerebelli below. All of that is dealt with by the ophthalmic division of the trigeminal nerve, and those primary nociceptive afferents, their central processes will then converge onto second-order neurons in the trigeminal cervical complex that are also being innervated by primary nociceptive afferents that have also come in from the trigeminal nerve, but which are coming from peripheral sites, hence why the pain is usually referred to the peripheral sites for the ophthalmic division of the trigeminal nerve. And what are those peripheral sites? They are the forehead region, they are the periorbital region around the eye, and they're the top of the scalp, the front portion of the scalp. And indeed, that is often where migraine pain is felt, because that covers such a large portion of cerebral cortex. Indeed, when you get the cortical spreading depression occurring in the occipital portion, which we know will then lead to the visual aura, that the pain from the activation of the overlying meningeal nociceptors, that we can understand is going to be referred to the ipsilateral, the same-sided ophthalmic area, so the forehead and the periorbital region, and I hope you understand that from our discussion of the innervation of the meninges. If it's then this lateral portion here of the meninges that's going to be activated, where is that going to refer to? Well, remember our discussion of the innervation. The front portion is going to be innervated by the middle meningeal nerve, a branch of the maxillary nerve, and it is generally referred to the anterior temporal region, the bit just alongside the eye, and maybe the cheek just underneath the eye, which is the peripheral sites of the maxillary nerve nociceptive innervation. And again, the basis for that referred pain is that the nociceptive afferent central processes from the meninges are going to converge onto the same second-order neurons that primary nociceptive afferents from the peripheral sites that are coming in the maxillary nerve are going to be synapsing onto. And then the brain interprets the activation of those second-order neurons as, oh, it was activated by the peripheral primary nociceptive afferent, i.e. it's coming from the peripheral area, not the meninges. And remember, the backward portion of this lateral area, that is innervated by the nervus spinosus from the mandibular nerve, and therefore, again, it's going to be referred to peripheral sites of the mandibular nerve's innervation, and that's that more posterior portion of the temporal region. Again, so that's why migraine pain usually is in the frontal, periorbital, the top of the cheek, and the temporal region on one side of the head. If it's actually occurring in the cerebellar cortex, then of course that will be activating the meninges over the top of the cerebellum, which are then innervated by primary nociceptive afferents that come in the posterior rami of the mixed cervical spinal nerves, C1, C2, and C3, and therefore it will end up being referred to the areas the peripheral areas that are also innervated by primary nociceptive afferents that are coming in the posterior rami of C1, C2, and C3, and of course that's the back of the head. So occasionally, some people do suffer from migraine headaches that are at the back of the head in the occipital region on one side again, and that might be because the cortical spreading depression is occurring in the cerebellar cortex rather than somewhere in the cerebral cortex. Okay, so we now hopefully understand the migraine headache and where it actually comes from, at least this story of where it comes from. And I'll remind you that this is just a theory. It's our best current theory, but it's a very satisfying theory, a very beautiful theory. Okay, so the next thing that I wanted to justify to you is why it's pulsating. We've done why it's unilateral, we've done why it's in the sites that it's usually in. Why is it usually pulsating in tune with the pulse? Why does the pain get worse and better, and it's worse when you've just had a heartbeat, i.e. the pressure's up in the blood vessels and they're all bulging, and then it's better for that instant where blood pressure's lower in diastole. Why is that? Well, of course, it's because most of the overlying meningeal nociceptors are surrounding arteries, both the extraaxial brain arteries that are in the subarachnoid space and the meningeal arteries that are in the outer meninges. That's the major site of meningeal nociceptors. Now, when the arteries bulge out, i.e. in systole, usually that does not activate those perivascular meningeal nociceptors. I mean, they'll be activated a little bit, but it isn't enough to actually cause the primary nociceptive afferent to fire an action potential. So imagine this is in the wall of some artery, and in fact you don't even need to imagine it. Let me draw an artery on here. So very simply, there is the tube, and there it is in the wall of this artery here. So let's say that this is a perivascular meningeal nociceptor around some either extraaxial brain artery or a meningeal artery. Okay, so usually when this thing bulges out during systole, when the heart has just beaten, that is not enough to cause this thing to fire action potentials. It will be activating nocicensors, which, remember, are the names for the actual receptors in the membrane of the nociceptor. It will be activating nocicensors in the nociceptor here, and therefore will be causing slight little depolarizations all over this nociceptor tree here, but they don't sum to a big enough depolarization at the beginning of the axon here to actually cause an action potential. So usually you do not get pain when your heart beats. However, when we have loads of prostaglandin E2 and loads of histamine and loads of the mediator from cortical spreading depression initially also activating the meningeal nociceptors, now every time the heart beats and the artery expands, again, what will be happening is those nocicensors that detect the mechanical movement, they will be being activated, but now their little depolarizations are going to contribute to all of the depolarizations that are coming from these other factors, and now they're actually going to make the depolarization here worse, and therefore they're going to make the action potentials that are firing more frequent. So what I am saying is that although normally they do not sum to enough to actually cause an action potential on themselves, when you have these other things that are activating the nociceptor and which are causing enough depolarization to cause action potentials to fire, when you add in these additional stimuli coming from the systolic blood vessel here, the bulged blood vessel, that's actually going to add a little bit onto the depolarization here, and it's therefore going to lead to more frequent action potentials firing. So what's actually going to be happening is every time the blood vessels bulge out, every systole, the action potential frequency firing along this primary nociceptive afferent peripheral process here is going to go up, and that will correspond to more severe pain. The more frequently action potentials are fired, the worse the pain is felt. So that is why the pain gets worse every time the heart beats. That is why the pain in a migraine headache is pulsating, because of the fact that most of the meningeal nociceptors are in the walls of arteries. Right, so now what I want to justify is some of the other accompanying symptoms of migraine headache. So we have done all of this. We've done moderate to severe headache, we've done pulsating, we've discussed the fact that it's unilateral, and we've discussed this site. This bit that I said earlier on about how women are 3 times more likely to be suffering from migraine than men, we don't understand why that is. Presumably the female brain for some reason is more susceptible to suffering from cortical spreading depression. We do not understand why that is. So I'm afraid I cannot justify that, like I cannot really tell you where cortical spreading depression comes from. Hopefully in the future these questions will be being answered, so we're going to live in a very exciting era. Okay, but what I do want to try and say a little bit more about is where the nausea and vomiting comes from, where these meningeal symptoms here come from, and where the cutaneous allodynia comes from, and then we will finish migraine. Or actually, no, we won't finish migraine, we'll talk about the treatment for migraine. So, nausea and vomiting, we understand that, remember, any pain can cause nausea, and then, if it's severe enough, vomiting, and we've discussed the reason for that. It's because Pathways that are not understood exist between the cerebral hemispheres and the vomiting center in the reticular formation of the brainstem, and when the cerebral hemispheres are experiencing pain, they can activate the vomiting center in the reticular formation, just as indeed the chemoreceptor trigger zone can activate the vomiting center if it detects horrible molecules in the bloodstream. And this can lead to nausea initially, but if the activation of the vomiting center becomes large enough, it can lead to vomiting. So the pathophysiology there is believed to be exactly the same as we discussed for tension-type headaches. So I don't have anything more to say there. Now, let's discuss the meningeal symptoms. Now, I cannot give you a particularly brilliant explanation for this because the particularly brilliant explanation does not actually exist. But I do just want to give you a little bit of motivation as to why these symptoms potentially exist. So, these symptoms are symptoms that always accompany headaches, well, usually accompany severe headaches that are originating from the activation of meningeal nociceptors. So when the pain is coming from the activation of meningeal nociceptors, it will lead to these accompanying symptoms. Whereas when we're discussing tension-type headaches, because the pain isn't believed to originate from meningeal nociceptors, they didn't have these accompanying symptoms. Now, we do not understand the mechanism of where these meningeal symptoms actually come from, and how they come about. And the main reason for that is we've discussed how there is convergence of the peripheral primary nociceptive afferents and the central, the meningeal primary nociceptive afferents onto second-order neurons. We've discussed in detail when we discussed referred pain how the brain couldn't actually tell the difference between pain coming from the meninges and pain coming from the peripheral tissues, and therefore it ended up referred to the peripheral tissues. That cannot be entirely true. Otherwise, how would these symptoms arise only when the meningeal nociceptors are activated and not when the peripheral nociceptors are activated? So there must be some way for the brain to distinguish pain that is coming from the activation of the meningeal nociceptors compared to pain that is coming from the activation of peripheral primary nociceptive afferents. However, we do not know what that mechanism is. So all I can tell you at the moment is that when you activate meningeal nociceptors, when the headache is coming from the activation of meningeal nociceptors, and you need a lot of activation of meningeal nociceptors, you will get these meningeal symptoms accompanying the headache. Now, I'll just remind you of what these meningeal symptoms are. Photophobia, intolerance to bright lights. Phonophobia, intolerance to loud noises. You don't like bright lights and you don't like loud noises. Knuckle rigidity, which is neck stiffness. So difficulty in moving your neck because the tone of the muscles in your neck has increased. They've all become very, very stiff and they're all contracting at once, so it's very difficult to move your head. And also the fact that the headache becomes worse with physical activity, so you can't really continue on trying to live your life normally because the headache just gets worse if you do that. So I can't justify how these actually work, but I can try and give you a little bit of motivation as to what these are trying to achieve. Why does the body have these? So if the meninges have been irritated, if the meningeal nociceptors have been activated, that means that there is something potentially very wrong inside the skull, very close to the brain, okay? So if the meninges have something wrong, it very possibly means that the brain has something wrong, that it's potentially a pathogen infecting these intracranial tissues, these very, very important tissues. And these are all to try and protect you, most likely. So knuckle rigidity is going to stop you moving your head. Making the headache worse with physical activity is going to mean that you want to lie down and not move your head. Again, both of these together are trying to persuade you not to move your head, so to avoid any further damage to the tissues within. If you don't move your head, then hopefully that's going to protect the tissues inside. Okay, so these two are both probably to try and protect you from damaging the intracranial tissues any further by moving your head. Why photophobia and phonophobia? Well, I can only sort of give you a concocted answer to this. It may well be very wrong, but I'm just to try and give you some motivation as to potentially why you might want these. Remember, if the meninges are damaged, it potentially means that the brain is damaged. And if the brain is damaged, then certain very important protective reflexes might not be working. For instance, the pupillary light reflex might not be working because this is a neurological reflex. If there's been brain damage, the pupillary light reflex could be damaged, and indeed, in many people who do suffer brain damage, the pupillary light reflex is lost. Now remember what the pupillary light reflex is. It's when you shine a bright light in someone's eye, the pupil constricts to reduce the amount of light coming into the eye and therefore protect the retina from the bright light, because the bright light could damage the retina. If the pupillary light reflex was damaged, as indeed if we've got meningeal irritation, there is a possibility that the brain could be damaged and therefore the pupillary light reflex could be damaged, then that won't be there anymore and there won't be anything to protect the retina from that bright light. So it would therefore make sense for you to be intolerant to bright light, for you to avoid bright lights. So that could be a motivation for why photophobia exists, because if the brain has been damaged, the pupillary light reflex could have been damaged, and therefore bright lights, the retina is no longer going to have its protective reflex against bright lights. Now, again, with phonophobia, there is a protective reflex to protect you from loud noises, and that's called the acoustic reflex. So remember, when very loud noises come into the ear, what happens is you contract the tensor tympani muscle and you contract the stapedius muscle, and those reduce the amount of the sound that is actually conveyed by the ossicular chain, the chain of the three ossicle bones, to the labyrinth, to the inner ear, to the cochlea. So less sound is actually going to be transmitted after the acoustic reflex. And again, that is to protect the inner ear. It's to protect the cochlea and the hair cells within the cochlea from the very loud sound, which could damage them. Again, this is a neurological reflex, and if there's been brain damage, which activation of the meningeal nociceptors potentially means there has, remember the brain doesn't have its own nociceptors, the meningeal nociceptors are the closest thing. If you've got activation of the meningeal nociceptors, that potentially means you've suffered brain damage, and therefore the acoustic reflex might not be working, and therefore you need something else to protect you from loud sounds, and hence you become intolerant to loud sounds, you avoid loud sounds, and you find them very, very not painful, but irritating, very irritating. Okay, so that is my attempt to give you some motivation as to why the meningeal symptoms might be a good idea to actually have when you've got activation of the meningeal nociceptors. However, I cannot tell you the mechanism of how you actually achieve these meningeal symptoms. It's not known. Right, so the final thing that I have to say in the story of the pathophysiology of migraine headaches is where does the cutaneous allodynia come from? So I'll remind you of what this means. So let's say that you suffer from very frequent right-sided migraine headaches, and you get it in the frontal region, so the forehead region, the right forehead region, the right periorbital region, and let's say the right temporal region as well. Okay, and you suffer from this very frequently, then what can end up happening is between each migraine attack, if you can have cutaneous allodynia in those regions. And this means that non-painful stimuli, non-noxious stimuli, stimuli that should not cause pain, will now cause pain if you activate the peripheral nociceptors. So if you touch the skin or press on the muscle, in those regions, it's going to cause pain. And this is a phenomenon that occurs in people who have very frequent migraine headaches, just as it occurs in people who have very frequent tension-type headaches. Now, this is more difficult to understand than in tension-type headaches, because in tension-type headaches, it made a lot of sense. You use those peripheral primary nociceptor afferents frequently when you're actually having the headache, because the headache originates from the peripheral nociceptors, and therefore the synapse in the trigeminocervical complex strengthens. But here it's more difficult because the headache is not originating from the activation of those peripheral nociceptors, it's originating from the activation of the meningeal nociceptors. However, remember, we have discussed convergence. There is convergence at the trigeminocervical complex. Peripheral primary nociceptive afferents, they synapse onto the same second-order neurons as the meningeal primary nociceptive afferents. And this, of course, is the basis for the referred pain. So let me just draw another little picture of this. So we'll put this little picture here. So let's say that this in here, this is representing the axon terminal or of the central process of, let's say, a peripheral one. So I'll just put a P here. So this is from a peripheral primary nociceptive afferent. And here, this is representing the axon terminal of the central process of a primary nociceptive afferent that is innervating the meninges. So this is the meningeal one. And they're both coming on to the same second-order neuron here. So here is the second-order neuron. Here are its dendrites. Remember, it's a multipolar neuron. And then here is its axon that will ascend up to the thalami, well, to the thalamus on the contralateral side, to the side that it's on currently. Right, so when you're having the frequent migraine attacks, what will of course be happening is the activation of the meningeal primary nociceptive afferent. So this one will continuously be being activated, and therefore what you would expect to happen is the sensitization of this synapse, and indeed that synapse does become sensitized. However, it appears that this second-order neuron here overall becomes more sensitive, i.e. it's not just this single synapse that becomes sensitive. The mechanism seems to be broader than that, so this sensitization seems to affect more synapses on this second-order neuron than just the synapse between this meningeal central process of this primary nociceptive afferent and the second-order neuron. So these synapses also seem to become more sensitive now. So mechanisms by which could underlie this, by which this could be achieved, is, remember, what probably happens is to increase the sensitivity of a synapse, you probably increase the number of receptors for the neurotransmitter that's being released by the central process here onto the second-order neuron. You probably increase the density of those receptors, and if this neuron's making more receptors, then they're probably going to end up being distributed all over the place on its dendrites. So in fact, it's most likely the case that a lot all over its dendrites, it's going to now become more sensitive to the neurotransmitter that's being released onto it. And therefore, what actually does end up happening is these other synapses become sensitized as well. So again, it's the convergence that underlies this. So that's a suggestion of how this could work, but it is a fundamental fact that you can get cutaneous allodynia, and it's in the region where it's referred to. Therefore, what must be happening is when you're continuously using this synapse between the meningeal nociceptive afferent and the second-order afferent, it seems that the synapse between the peripheral afferent and the second-order afferents also become strengthened, and therefore when you apply a stimulus to the peripheral area that should not cause pain, what that would lead to normally is maybe some action potentials coming up this peripheral neuron here, but previously they wouldn't have been enough to actually activate this second-order neuron, but after the sensitization of this synapse, now those action potentials are enough to activate the second-order neuron, and therefore will result in the second-order neuron firing and pain. So that is believed to be how cutaneous allodynia works in migraine headache, that you don't just get strengthening of the specific synapse that is being used hugely in the trigeminal cervical complex, but you get sensitization of the entire second-order neuron, and therefore other synapses onto that same second-order neuron that are coming from the peripheral nociceptive afferents will also become strengthened, and therefore stimuli that would not have previously actually made it across this synapse, wouldn't have previously been able to activate the second-order neuron. Action potential frequencies that wouldn't have been able to activate the second-order neuron are now capable of activating it, and hence how you can get cutaneous allodynia. Okay, so that is my discussion of the pathophysiology of migraine headaches complete. In the next video we will move on to the discussion of the treatment for migraine headaches, and again we'll split this into abortive treatments and prophylactic treatments.